Well, it's a delight to be here again. My name is Susan Fox. I'm the Associate Director of the clinic. Tony Lang, the Director, is off at a talk in Kingston, so sends his apologies. So, welcome everybody. This is the 7th edition of the Movement Disorder Speaker Series. I can't believe we're now in our 7th edition. This has been a real success and hopefully will continue to be so. These speaker series are really only possible with the very generous help and donation from donors, and particularly this session has been made possible by Fraser Berrill, who's here today. Thank you very much for your support for the series today. Thank you. So for those of you who have been here at the previous series, each of these series is really given by a member of the faculty, and the idea really is to inform you of what we're doing, what's going on in the world of research into movement disorders, and to allow us to tell you what's happening and invite you to ask questions. And so hopefully everybody goes away with a little bit more knowledge and understanding. The session today is going to be led by Dr. Slow. Dr. Slow is one of our faculty. She's a clinician teacher. And she's going to give you an overview on genetics in movement disorders, including Parkinson's disease and dystonia. Dr. Slow.
Thanks so much, Susan, and thank you so much for having me today. This is a very exciting topic, and without further ado, so I'm going to be talking today about genetics in Parkinson's disease and other movement disorders, and certainly the field of genetics is growing exponentially, and the importance of genetics in medicine, including the medicine of movement disorders, is increasingly being recognized, and that's what I want to touch on today. And just a little bit of a history lesson. This was the original photo that Rosalind Franklin made in 1952, which led to Watson and Crick discovering that DNA was a double helix. So it was really this picture that led to our understanding that the DNA molecule, and I'm going to talk a lot more about that, is in this very particular structure, which led to their Nobel Prize in 1962. So I'm going to structure my talk first by really talking about DNA and genetics and introduction. Some of you may have a lot of background in this, some of you might not have very much, so we'll go right from the beginning. And then I want to do a demonstration of genetics with the whole audience, if you're open to it. Then I'm going to talk specifically about genetics in Parkinson's disease, and I'll touch on some of the other movement disorders that we see in the clinic and the genetics in those disorders. And that part of the talk will be about our current understanding. And then I want to finish the talk with talking about genetics in Parkinson's disease and other movement disorders, the future. So what are the possibilities for genetics in movement disorders? Okay, so first of all, what is DNA? So DNA is a molecule which carries the genetic instructions which is used in the growth, development, functioning, and reproduction of all living organisms, and of course, very importantly, humans. And so what DNA looks like is it's this, as I said, this double helix. And the DNA, when you, one unit of coding information is what we call a gene. All of this DNA is wrapped up very, very tightly into what we call chromosomes, and humans have 46 chromosomes. And then these chromosomes are even more tightly wrapped up into the nucleus of our cell. So every cell in our body has this DNA code within it. And so we can talk about DNA, make the analogy to an alphabet. So of course we have our alphabet with 26 letters. DNA alphabet is only 4 letters, so C, T, G, and A. A group of these letters together that has meaning is called a gene. And we can think about that similar to a sentence, so genes can be very, very long, so it'd be almost like a run-on sentence in many respects. Many, many genes put together makes up a chromosome, and so we can think about that like a chapter of a book, lots of information. And then when we put all those chapters together, so 46 chapters, then we have a book, and all of those chromosomes together again make up the nucleus of our cells, so all of that genetic information. So genes have all this information, but what do they do with it? And so what genes do is they make proteins, and proteins are the building blocks of our cells, they're the building blocks of our body, they're signaling molecules to tell our different body parts what to do. And of course, we don't just have one gene, we have many, many, many genes encoding for many, many different proteins. And together, all of this makes up a human person. So then I'm going to talk a little bit about the human genome. So the human genome is the complete set of DNA. And so this was something that we didn't know what all those letters were. We didn't know what the sequence was until the early 2000s when the whole genome, so that complete set, was finally discovered. And the total length is 6 billion letters. So if you think about that book analogy, we would need 2.6 million pages in that book to encompass all of the information in our DNA. So huge amount of information. And it's organized into 22 paired chromosomes. And so this shows a diagram of all of those chromosomes. They come in pairs. And then if we're female, we have 2 X chromosomes. If we're a male, we have an X and a Y chromosome, and that makes 23 pairs of chromosomes in total, so 46 chromosomes. Now, interestingly, before the genome was completely sequenced, so before we knew what all those letters were, we thought there were many, many thousands, hundreds of thousands of genes, but we actually found that there are only 20,000 protein coding genes. And interestingly, all humans, so everyone here, shares 99.8% of our DNA. So we're all, which shows us we're all actually very, very similar, right? There's only 0.2% that separates one person from another person. So why are we different then? Well, variety is the spice of life, and so variety or variation in genetics gives us the differences that we see from person to person. So genetic variation are little changes in these letters, little changes in the letters in these genes, gives us the differences which makes us unique. And you can see in this picture all these different skin tones, hair colors, eye colors, so all of those different variation that we see from person to person. Now, of course, variety is not always a good thing. There are what we call pathogenic, so disease-causing variation. And so those are differences in the DNA which cause disease. These are often rare, and the first genetic disorder that we knew the gene for was a disorder called cystic fibrosis. And this is a disorder that affects the lungs, usually of children, very young age. And then DNA, you can always blame your parents because your DNA comes from your parents. We have 2 sets, 2 copies of each gene, one from our father, one from our mother. And that's how we get this almost infinite variation. Okay, then I want to talk a little bit about the differences in how we get variation. So we can have one gene that variation in one gene causes differences, or we can have many, many, many different genes, and of course, and the environment playing a role. That's what we've referred to as monogenic, with one gene, or multifactorial, many genes, all the environment. And we can talk about traits. So for example, eye color. So eye color is caused by changes in one gene, whereas height is caused by changes in multiple genes as well as the environment that you're exposed to. And I thought this was interesting. These are Hollywood leading men and the differences in their height. And you can see, so here's Tom Cruise, who's this action hero, and he's of course on the lower end. I think sometimes they even put him on a box sometimes in his movies, so he appears to be this towering presence. And then we can also, of course, see this in diseases. So I already mentioned cystic fibrosis is a one gene disease. But diabetes, we think diabetes is caused by changes in multiple genes, but also we recognize the environment is very important. As obesity rises, we know that diabetes equally rises, and we know there's a relationship between the two. Okay, so what I would like us to do is to look at genetic variation in this audience. Now, we're not talking about disease, we're just talking about little trait variation. And so actually, so Steph, if you could hand out some of these. So you're gonna get some papers, and I don't want you to do anything with them yet. This is something you can try to taste in a few minutes, but I'll talk about it. But before we get there, so can you roll your tongue? So I see some people already rolling their tongue. So I cannot. So and this is not something you can teach yourself to do. No matter how much I try to roll my tongue, I will never be able to roll my tongue. This is caused by a change in your gene. Why? Why some people can roll their tongue? It doesn't seem very useful to me. But so about 70% of people can roll their tongue, and about 30% are like myself. I can't do anything with it. Rollers, oh, we got a rolling, there you go, 70%, very good. All right. Any rollers over here? No, some rollers? Rollers, good. All right. Now, this you may need a friend to help you determine, is your earlobe attached? So, and by attached, it means that it goes right, the skin goes right, the earlobe attaches directly to the skin of your face, or it sort of dangles here, right? So I have dangly earlobes. Dangly. Anyone dangly earlobes? And again, you may need help with this one. You may need someone to take a look for you. So 75% are unattached, like myself, dangly. 25% are attached. And again, we think this is likely due to one gene, maybe a couple of genes that play a role in this. And why? Who knows, right? It's not very useful either way. Now, can you see this? So red-green colorblind, fairly common. So 1 in 12 males, 1 in 250 females. And that's because the gene that causes this change is on the X chromosome. Right, and males only have one of those, so they're more likely to be affected. So can you see this number? So can anyone see that number there? It's a little fuzzy, but that's a 45. And so if you can see that, you can likely see colors, no? Don't be alarmed. It's okay, it's pretty hard on the screen. Yeah, this is challenging on the screen. And so this is just to show what someone with red-green color blindness would see. So they wouldn't really see any green or blues. They'd just see a lot of reds or oranges. And similarly, if they looked at a painting like this, so this is the painting most of us would see, all these vibrant colors, and there, it would be a bit more washed out. So no blues, those greens wouldn't be as vibrant. And again, usually doesn't cause any problems with living a very normal life. Okay, and then finally, what I'd like you to do is see, okay, so I've given you a little piece of paper, hold on. This is something that apparently has a terrible taste. So I suggest you only just dab it at the tip of your tongue. I can't taste a thing. So C, anyone? Yes? Oh, see? Sorry. Oh no. Okay. So that's are you okay? Okay. Can you taste? I think there's some people coughing, so I think they can taste it. Sorry about that. So, can people taste it? Anyone? Anyone taste it? Taste it? Taste it? Okay, there we go. So this is something called PTC. It's a chemical. It's not in any of our foods. Some people can taste it very strongly. I saw this poor gentleman make a wincing. And of course, and then a lot of us, nothing, right? And again, this is thought to be due to one gene. Not very useful, but there it is. So those are some examples of some simple genetic traits. All right. So now we're going to move on to talk about genetics in Parkinson's disease. Okay, so 90% of Parkinson's disease is thought to be sporadic. What does that mean? It means we don't know what causes the Parkinson's disease. It doesn't seem to be running in families in those patients. And this is compared to 10%, which is genetic. And genetic Parkinson's disease is caused by changes in genes, and we're talking about single genes, one gene, which we know will cause Parkinson's disease. So only 10%. And so what are some of the genetic or single gene Parkinson's disease? So I'm just going to talk about a few here. So in some patients who have early onset Parkinson's disease, that means they start to have symptoms sometimes in their 40s, sometimes even 30s or 20s. Two of the genes we see, one of them is called SNCA, and this is a gene that makes the protein called alpha-synuclein. And I think a lot of you in the room will have heard of alpha-synuclein, and we'll talk about that a bit more. Another of the early onset genetic causes is something called Parkin, so a gene called Parkin, and we found that that is the most common genetic cause of early onset Parkinson's disease. But again, both of these are quite rare. Now, later onset Parkinson's disease, and so that's when most people get Parkinson's disease, the most common cause is something called LRRK2. And again, you may have heard of LRRK2 before. And this has been found in 1% of people with PD have mutations in this gene, changes in LRRK2 that are causative for their Parkinson's disease. And just so you understand a little bit about the complexity of this, we know that there are 6 genes for classical Parkinson's disease, so that look like Parkinson's disease, and then 17 other genetic causes of what looks like Parkinson's disease, but actually has a lot of other features which makes us think maybe that's not Parkinson's disease. So a lot of genes at play. Now, just to highlight, so Lorraine Kalia, who has presented at these rounds, she's one of our faculty. She is an expert in alpha-synuclein, so she works on alpha-synuclein and very interested in genetics as well and being involved in genetic research. And then Connie Maris is one of the world experts in LRRK2. And so she does a lot of work in LRRK2 and is also an expert in genetic causes of movement disorders. All right, so those are rare disorders, but how can gene discovery impact Parkinson's disease knowledge overall? And so one way it can is by, as I said, increasing knowledge about why Parkinson's disease develops in general. So not just the genetic, but all Parkinson's disease. For example, this gene I talked about, SNCA, which makes the protein alpha-synuclein, was the first genetic cause of PD discovered in 1997. It's a very rare cause. But it led to the discovery that alpha-synuclein is a major component of Lewy bodies. And Lewy bodies are what we see in brains of all patients with Parkinson's disease. So this has led to a lot of research into alpha-synuclein and the potential role that alpha-synuclein plays in causing Parkinson's disease, and potentially how to try and target that to slow down Parkinson's disease. Now, what about the other 90% of PD? So the so-called sporadic, the PD we don't know why people get PD. So we think that it's likely a mixture of genetics and environment and the interaction between. And I'm not talking about a single gene, but variations in multiple genes. And so if we look at a scale, if we have people with PD, they likely have subtle changes in multiple genes that put them at higher risk of developing PD, and then in addition may have some environmental factors that have also put them at higher risk. For example, pesticides, head injuries. On the flip side, there are genetic changes, variations in certain genes that seem to be protective against PD, and then also environmental environmental things that can also decrease the risk. For example, there's this, we know that people that smoke don't get Parkinson's disease at the same rate for some reason. We certainly don't advocate smoking to prevent Parkinson's disease, but what is that? And also coffee. So, and this is just, again, to show you that, so it seems that, again, caffeine decreases the risk. Again, these are associations. We don't really know what they mean. It's not a specific cause and effect. But I can get back to that, yeah. And again, to show you the complexity, this is something that Lorraine Kalia and Dr. Lang put together. So just listing potential genetic risk genes that could increase your risk. Changes in these genes could increase your risk. Changes in these genes may decrease your risk. Changes or these environmental risk factors may decrease your risk, and these ones may increase your risk. And again, we don't know exactly what the relationship is here, but this is the hypothesis as to why most people get Parkinson's disease. So I'm just going to talk about a few of the other movement problems we see in the movement disorder clinic, because genetics are a huge component of those disorders as well. So we see patients with dystonia. So dystonia is abnormal postures caused by involuntary muscle contractions. And you can see in this statue has an abnormal position of their head. And in dystonia, so we remember Parkinson's disease, maybe 6 plus 17. In dystonia, there are 30 plus genetic causes. We used to think most of the genetic causes were in younger patients, but now we're recognizing even older patients with dystonia. And in dystonia, if we have a genetic cause, that can sometimes help guide our treatment. We also see a lot of patients with ataxia. So ataxia is a problem with coordination and balance. Very different than the balance problem we see in Parkinson's disease. Now, again, getting more complicated, 122 plus known genetic causes, but there are many, many unknowns about ataxia, so causes of ataxia. This is just information from our clinic. If we look at all the patients that come with ataxia, we find over a quarter of them are genetic. About an eighth of them have something called multiple system atrophy, which can look like Parkinson's disease. But over 50%, we don't know the reason. So we still are struggling with what causes ataxia. And currently, there aren't any treatments for ataxia. So in Parkinson's disease, we do have levodopa, which can help with the symptoms. Unfortunately, in ataxia, we have no medications to help with the symptoms. And so Dr. Munoz and I are establishing an ataxia clinic to try and better understand, diagnose, and in the future, hopefully, treat patients with ataxia. Okay, so who do we test for genetic causes? So if patients have a very strong family history, and this is why we ask about family history when you first come to visit us. And so this is what's called a pedigree or a family tree. The males are squares, the females are circles, and you can see this is one generation with their children, and then this generation has also had children. If someone has a disease, then that's denoted by having a dark coloration. And so if you saw a family tree like this, where almost half the individuals are affected, then that's a very strong clue that there's a genetic cause. Patients with onset at a young age, for example, patients with Parkinson's disease that have onset in their 40s and their 30s, very increases the probability that we're dealing with a genetic cause. And then also patients in high-risk populations. We know certain ethnic populations or certain ethnic backgrounds have a much higher risk of certain types of genetic disorders, and in those populations we may test. Okay, and then genetic testing, well, some people say, why would we test if in most cases there is no treatment? And really, even if we have a genetic diagnosis, that is in most cases the truth. We don't have a specific treatment to offer. But it can answer that question, why do I have this disease? And for some people, it's very, very important to understand why do I have this? A genetic diagnosis can also help with prognosis. So people often say, what is my disease going to look like in 10 years? And this can help. For example, that Parkin type of Parkinson's disease I talked about, that genetic cause, tends to have a much slower progression. Patients get their disease in their 20s and 30s, but over time it progresses much more slowly. And then for certain diseases, it can have implications for family planning and other family members. Less so in Parkinson's disease, but for example, in Huntington disease, which is 100% genetic, people will often reconsider perhaps having children if they know that they're carrying the gene that causes Huntington disease. All right, and so what I'm going to do is conclude the talk with talking about genetics and the future of genetics. And so hopefully some people recognize that this is the future, the enterprise, original enterprise. And so this is, again, what I was just talking about. Those are things we do currently in the clinic. We test certain people for genetic causes, and there's a lot of research into those genes and what they do. But now what I want to talk about is the future. Okay, so potentially in the future, gene discovery may help with prognosis. So often people say, what type of PD can I expect? So some people have a tremor that's very predominant. Other people may have difficulties with falls or balance earlier in their disease. And we think, again, that there are changes in different genes that lead to these different changes. And if we knew what those changes were and could look at them, then we could let people know what to expect. Another big question is, what other problems can I expect in my disease and when? And so, and a lot of people especially want to know memory and thinking. Is my memory going to be affected and when? And again, we think that this is likely a combination of factors, subtle changes in a variety of genes, environmental factors, which together lead to this. And there's been a lot of work trying to predict using different algorithms, looking at genetic changes, environmental changes to try and predict in populations. And so far, we're not very good at those predictions, but hopefully with more information, we'll get much better. And then finally, what treatment will work for me and will I have side effects? And this gets into an area called pharmacogenomics. And so I'll move and talk a little bit about that. So pharmacogenomics is talking about how genes impact how drugs work. And so you can imagine that we have this patient population, and we think they all have the same condition. So everyone, has, in this group, we can imagine they all have Parkinson's disease. Now, at the moment, what we do is we treat everyone with levodopa, right? And we see, do people respond? And a lot of people respond well, good response. Some people don't have any response, and some people have very poor side effects. But until we treat, we don't know. Now, pharmacogenomics suggests that there are changes in genes that can predict this. And so what we would do is take this population, test their genetics, and then be able to tell which of that group of patients would respond to the drug and only treat them. And then again, thinking about if you had multiple drugs, then you could do this for multiple drugs to find out what the best combination of drugs would be for each patient. And again, just to highlight some of the research being done here, so this is Alfonso Fasano, who is one of our DBS movement disorder specialists, and he's looking at pharmacogenomics in DBS to see if they can predict response to deep brain stimulation. And then again, so how can gene discovery impact my disease? So treatment, and of course we're always hoping for new treatments for Parkinson's disease and other genetic disorders. In Parkinson's disease, so alpha-synuclein, so I talked about that a number of times now. So alpha-synuclein has been targeted for treatment since it's thought to be part of the disease, so part of the pathogenesis of Parkinson's disease. And so this is being targeted now with antibodies. So antibodies break down proteins and get rid of proteins that we don't want. And so the thought with these clinical trials is to try and decrease the alpha-synuclein and then hopefully slow the progression of the disease. And at the moment, there are three different antibody clinical trials. GBA is a gene I haven't mentioned yet. So GBA, we know that changes in this gene increase your risk for Parkinson's disease. They don't cause Parkinson's disease, but they seem to increase your risk. And at the moment, there are three different compounds targeting the GBA pathway that are in clinical trials. And then finally, in LRRK2, so LRRK2 being that very prominent genetic cause of Parkinson's disease, they're now looking at inhibitors of LRRK2, and it seems to be well tolerated, and it's moving into the next phase of clinical trials. And these are all based on genetic discoveries. And this moves towards this paradigm shift in how we treat all diseases, going from this one-size-fits-all medicine. So this is what I spoke about before. So we have this group of patients. We give them all the same therapy, and then we find that some people have adverse events, some people don't have any benefit, and then a few people have benefit. And trying to take this, study the disease, looking at different disease subtypes, different clinical features, maybe some biomarkers, and then look at the genomics, looking at the genetic background, using this and then coming up with very specific or personalized medicine. So certain drugs for each of these different populations. So that's what people are moving towards in cancer, and that's what we're hoping to move towards in treatment of movement disorders as well. And this is what this potentially could look like. So we know that there's something called prodromal PD. So the disease starts years before the symptoms are first present. And so if we could figure out who these patients are, look at their genetic background, then if they have this GBA mutation, they could be treated with GBA-specific therapies. If they have LRRK2 mutations, they can be treated with LRRK2-specific therapies. And then when they develop Parkinson's disease, again, we can look at, currently we only have levodopa and levodopa-like medications as well as our surgical indications, but look at these through pharmacogenomics to try and figure out who of these patients we need to treat with these different strategies. And then, of course, on the other side, we have all sorts of non-motor problems, so cognition, fatigue, sleep, and again, use personalized medicine strategies to try and treat these different disorders. And you can imagine if we do find those disease-modifying therapies, they may be able to slot in up here so that the clinical course of these things changes as well. And then, of course, in personalized medicine, we have to take into account age, lifestyle, comorbidities, all sorts of different things. So really the whole picture, instead of just treating everyone the same way. Oops. Okay. And so I'm just going to end with so this is the group, and I think if you've been coming to these, you've seen every one of us now. So, of course, this is Dr. Tony Lang, and he's been instrumental in many gene discoveries and movement disorders throughout the years. And then I talked about, of course, Connie and Lorraine. And then I'll leave it up for questions. And so this is a chimpanzee, and interestingly, genetically, we're 99% the same as chimpanzees. So maybe that, so that's interesting of itself. So, and then again, I'd like to thank very much Mr. Berrill for supporting this. So thank you so much.
We'll open the floor to questions. We've got plenty of time. And, okay.
I do have a question. Has Parkinson's disease been identified in any other species other than human animals?
No, not to my knowledge. I don't know. No.
I don't think anyone
has looked.
Animal, not looked. I don't think anybody has looked, but we're the longest living species, and I think we know that age is such a big risk factor. So even in elderly primates that live for 30, 40 years, I don't believe anybody's discovered the same pathology. Oh, I'm sorry.
Yes.
When you talk about the clinical trials, how extensive, how deep?
Sorry, what?
Just how big is it when you talk about a clinical trial? How extensive?
So how many patients are involved? Yeah.
Yes.
So it's highly dependent on the clinical trial. So some of the antibody trials that I'm talking about have 200 patients enrolled in them. But again, it very much depends on the clinical trial how many patients they enroll, how many patients they think they need in order to see a significant effect to say whether the drug they're trying is effective or not. And then another aspect of clinical trials, of course, is how long. So clinical trials can be many at least months, but some of these clinical trials, there's a thought that maybe we're not looking long enough to see effects. And so some of these newer clinical trials, for example, some of the antibody trials are going out to 2 years to see if they can find a clinically significant effect. With interim analysis, of course, before now. Dr. Fox is a clinical trials expert, so maybe she can add to that.
Yes, I think it depends which trial we're referring to, but the genetic trials are probably going to be fairly long trials. The alpha-synuclein antibody trials have already started. We will be a site, we hope, and eligibility criteria will obviously determine if people are able to take part in the trials, but they're certainly going to be happening, and there'll be more and more coming up in the future as well. So there's the alpha-synuclein, they've gone through phase 2A, so that this will be sort of what's called a 2B. Yeah, so they're still not very, very large, but they're getting bigger.
How is this information, like, this is your own research?
No. So these clinical trials, the ones I was talking about, these are pharmaceutical sponsored trials. Yeah, these are companies, and they're multi-center companies, they're company, but then multi-center studies. So with groups around the world. So we're, as Dr. Fox is mentioning, we're a single center, but they're multiple centers. Yeah. Okay.
Could you elaborate a little bit more on the nature of the antibodies that would be used to combat alpha-synuclein? Whether the nature of the drug and if it's a repurposed drug or...
Yeah, so certainly. So these are not repurposed drugs. So these are antibodies that have been developed specifically against alpha-synuclein. So not thought to have any other role. They've been developed specifically to trial treatment in Parkinson disease. They are, the type of drug they are is infusion drugs. And so that tends to be what antibody trials are. So it's a once monthly infusion of an antibody and then follow up. And there's been a lot of history of antibody research in Alzheimer's disease of sort of varying success. But this is certainly a place where we're going in neurodegenerative diseases, because we realize that in many of our neurodegenerative diseases there are particular protein problems. In Parkinson disease, it's alpha-synuclein.
Hi, my husband has Parkinson, and he's diagnosed with a Parkinson dementia. And there's nobody in the family who has Parkinson, but the mother has Alzheimer. Is it related to any genetic in the family or something?
Yeah, so it's a very good question. So we think that Parkinson disease and Alzheimer's disease are very different diseases. So if it's indeed someone had Alzheimer's disease and someone else in the family had Parkinson disease, then it's unlikely that the two have anything to do with each other and are genetic.
He's having a Parkinson dementia. Is it anything to do with the Lewy body in the brain that you're talking? And is there a difference having a Parkinson dementia, which I don't really know what is it, and some kind of other dementia?
Yeah, so that's a very good question, and certainly a lot to that question. So first of all, so what does it have to do with Lewy bodies? So we think in Parkinson disease, what happens with Lewy bodies is that they start out in the brainstem. And specifically the area that's affected in the brainstem is an area called the substantia nigra, which causes the slowness, the stiffness, the tremor that people present with Parkinson disease. However, over time, we think that those Lewy bodies move throughout the brain. And again, very different in different people, but in some people, those Lewy bodies will move to places that then cause memory problems. So that's the relationship between Parkinson's disease and dementia. When they move to those areas of the brain that can cause memory problems, then you can get dementia from your Parkinson's disease. But unrelated to Alzheimer's disease, which is very different, no Lewy bodies in Alzheimer's disease, a very different protein problem there. Yeah.
I have two questions about likelihood.
Mm-hmm.
You talked about inheriting a gene from your parents and grandparents.
Mm-hmm.
If I have 3 adult children and 5 grandchildren, what is the likelihood of my having passed on any of my children? And if so, what is the likelihood of them actually getting Parkinson's disease from that gene that they've inherited?
So what I would say is it's most likely that you have what we call sporadic Parkinson's disease. And in that case, they wouldn't be at an increased risk for developing Parkinson's disease. However, as we know, Parkinson's disease is fairly common, so 1% at the age of 65, and that increases as people age. So they have that background risk, but not an increased risk.
I've read recently that there might be an association between risk of developing Parkinson's and presence of autism spectrum disorder. Is anyone in the Toronto General Clinic looking at that association?
So no one in the clinic is looking at that. I'm not sure what that particular study refers to. I don't know if you know that one. Yeah, I haven't seen that. Yeah.
Thank you for your great talk. Just wondering how you're counseling people with early onset Parkinson's about doing genetic testing, specifically related to the implications for their children, so that kind of early onset group, and what you're doing in terms of that counseling right now.
So genetic counseling is certainly very important. In any time we test someone for genetics. So we have to let people know that if there is a positive result, it could have implications for their family members. Of course, some of these disorders, especially the early onset disorders, you need 2 abnormal copies in order to have the disease. So one from mom, one from dad. So that means they would have to have a spouse that also has an abnormal copy in order to have their children be at risk. So it also depends a bit on what the genetic results are as to whether there's an increased risk for their children of inheriting that. So we talk about that, and then we also talk about the possibilities of what comes back from the genetic testing. And and of course that's up to the person whether they want to go through with the genetic testing, and if they don't at the time, then we can always bring it up later if they would like to think about it. So it's an option that's given to them.
I have a hobby of my family tree. And at the moment, it's over 1,000 people.
Oh, fantastic.
There's nobody.
Oh, we love that.
Interestingly enough, the Parkinson has not raised its head at all, except for me.
Right.
And my brother. And in both cases, it was in their late 80s before it developed.
Yeah.
So is there anything significant in that?
Yeah. Significant. Yeah. So Dr. Fox just mentioned there's probably a lot of protective genetics within your family to have so few family members affected, and for you and your brother, of course, fortunately to have the disease, but to have it much later in your life. So not likely a genetic cause for your disorder. Well, I mean, that's very, That's a very good point, right? If people only live to 40 or 50, then the prevalence is much, much decreased, right? So that is a very good point, yeah. 3 brothers, yeah. Wow. Yeah. Amazing. Yep. Mm-hmm.
Yeah, just curious on how the change has gone from medication to closed-loop DBS. Have we seen like a lot of higher results compared from medication to DBS?
So are you talking about medications versus DBS?
Yes.
So, so DBS is a very specific therapy, and often we use that therapy for patients that respond very well to medications, right? So that's actually one of the criteria for most patients with PD who undergo DBS is they have to respond well to the medication, but they're in a part of their disease where the medication is causing lots of side effects. So either dyskinesias, extra movements, or these offs and ons, so these motor fluctuations. Those patients tend to be very good candidates for DBS. So, and often patients, even after DBS, are still on levodopa. It may be able to be reduced, but they still need the medication. So.
So just with respect to clinical trials, can you tell us how many clinical trials are currently taking place here at Toronto Western or at UHN, and what makes us the ideal kind of center for these trials to take place?
Sure. Okay. Well, that's a good question. We do a lot of clinical trials. I was lots of samples from people, so collecting samples from large groups of patients, people, family members is something else that we do, and we collaborate with other centres in Canada and other parts of the world. So there's a whole range of studies going on. To be an eligible candidate for a study depends certainly on what type of condition you have, what medications you're on. There are lots of criteria. Many of the studies are open to anybody because we're just really wanting simple information. Others are very specific. So, for example, the infusion trials with the alpha-synuclein studies, those are going to be very specific people that would be eligible. Others is really open. We have a list of our clinical trials in our clinic. People who are interested can just ask their physician when they come to clinic. We flag every single person that comes to the clinic, and we have an identification on the front of the chart, and if that person, that the clinical trials coordinators have gone through and seen if that person is eligible, and then it will be discussed with the person when they come to clinic. So we have several ways of trying to identify people who may be suitable, but it's also, people can always ask, and we'll obviously let people know what's going on. We're always looking for people to take part in trials. Yeah.
Just further to that, can you just talk about overlap with other specialty areas in medicine? So, for example, in ophthalmology, there are studies that are they're looking at Alzheimer's detection through eye tests. And so is there examples of that with Parkinson's as well?
So, well, there have been and there are ongoing studies trying to find what we call biomarkers that might help us detect conditions better, such as Parkinson. So, obviously, brain scans, brain imaging, skin biopsies, bowel tests, bowel biopsies. So, yes, we do interact with other specialities, gastroenterology, radiology, to try and improve. And some of these trials do involve other specialities. That's true, yes, yeah. How are we doing for time, Steph? 5 more minutes, okay.
Okay, I have a question about, that relates directly to me about hand dystonia, writer's cramp, and what's being done on that, and what type of remedies there might be to, if there are any, to try and correct that or improve that situation.
Yeah, so I'll answer that quickly, and then we can always talk after as well. Dr. Robert Chen is one of our, so here, he's actually doing a study on focal hand dystonia, and he's one of our experts, as is Dr. Fox on dystonia. And then really treatment of focal, so hand dystonia, we often use something called botulinum toxin. But as I said, we can certainly discuss that further afterwards. Yeah. Yeah, go ahead.
Hi, I have a question about more clinical trials and research studies in general. Often the upper age limit for prospects is 80, sometimes 85, rarely as old as 90. I wondered why that was. Is it because patients that old are considered too old to live, or their bodies will react in an entirely different way, or
Right.
what is generally the reason?
I think, so for many of these trials, what they want to do is get as homogeneous, which means as similar a population as possible. Of course, I mean, we recognize that Parkinson's disease, for example, is very heterogeneous, not that homogeneous. But in some of the ways they do that is by limiting the age. So, for example, between 40 and 80, so trying to get rid of those very young onset, and then also the elderly population. And it could also be because they want to limit what we call adverse events. The pharmaceutical company wants to see that their drug works, and if people are having falls for other reason, having difficulties for other reasons, which may be blamed on their drugs, then they want to try to not have that happen. So it doesn't mean that the drug wouldn't, if it proved to be useful, wouldn't be used in someone who has Parkinson's disease over a certain age. That wouldn't be a limiting factor. I don't know if you want to add anything to that. So it really depends on the research study, yeah. So as Dr. Fox was saying, we have many, many different research studies. We're trying to find out more about Parkinson's disease in general, and so that's for everybody, all comers. And then some of these studies about the biology of Parkinson's disease, again, all comers. It's really more of those pharmaceutical trials where they really want to try and limit things, and they have a lot of exclusion criteria because they really just want to see if their drug works. Yeah, sort of single focus, and that can be a problem with drug trials if the population is so focused. Well, even if it works, then how generalizable is it, right? But this is what they do just to get that first step, right? Does it work? Mm-hmm.
Yes, we are. We're doing what's called an open label study, which is administering a drug that's already available on the market for people with cervical dystonia to see if it's safe and tolerable and there's not adverse effects. And if it shows what we call tolerability, then the plan will be to do a standard double-blind randomized trial. So yes, we are. And we're hoping next year there is a new preparation of the botulinum toxin that will also be part of that trial, which is a pharmaceutical trial. But the other trial is one that we've initiated in our clinic. So yes, we are.
Okay. And so,
No, fine.
So I've got the signal that we're going to have to wrap it up, but I'll certainly be available for a little bit longer if people have any more questions. But just wanted to thank everyone for coming out, and again, thank you so much for sponsoring this. It's been great. Thank you.